Now this provirus is a permanent part of you. It gets integrated, and as I say, we don't know how to take it out. The virus doesn't want to take it out. It just, the way the virus gets out of the cell that has the provirus is by having the cell make mRNAs, and those get packaged into new virus particles. So the only way out of the genome is by transcription. Now when the provirus goes into your germline, which it can do, these are called endogenous retroviruses. So you can imagine that if you get infected by a retrovirus, it could just infect skin cells or respiratory cells. It's not much of a consequence. But if it gets integrated into your germline, then you can pass it on to others. And this is why I said initially at the first lecture, 8% of our DNA is retroviral because we have a whole bunch of these endogenous retroviral genomes in our DNA. These are remnants of being infected many, many years ago. And these are called retroelements. These are retrovirus-related sequences, and as I say, endogenous retroviruses or endogenous proviruses are in the germline as opposed to a somatic cell. The ones we have are replication defective. We don't make any retroviruses because all of our retroelements are defective. They have mutations in them that prevent viruses from being made. But about half of our genome is composed of mobile elements that are related to these retroviral sequences, including endogenous proviruses, but there are other retroelements as well. And this table summarizes that. These are different kinds of retroelements that are in us, in our genomes. So for example, endogenous retroviruses, so that's the proviral DNA sitting in our genome. That's about 8% of our DNA, old infections that happened probably millions of years ago, and these DNAs are still with us. There's another kind of element called a retrotransposon, which is basically the same as an endogenous retrovirus, but it's missing the envelope glycoprotein. And you can see in all of these diagrams we have cellular DNA in purple. It's duplicated on either side. Then we can have LTRs, and then some of the viral genes are shown here. About 20% of our genome is comprised of what are called line elements. And these again are retroelements. They have duplicated host sequences. There's no LTRs, and the sequences here are retroviral, but they're completely mutated and don't express retroviruses. Some of them do make reverse transcriptase. There's one particular line element in human called L1 that actually does make reverse transcriptase. So all of us have a very low level of RT in some of our cells. And the consequence of that is that other mRNAs can be converted to DNA copies. So for example, the signs are another kind of retro sequence. You can see duplicated sequences at either end suggesting integration by RNase H, but these don't have very much retrovirus homology. They certainly have no RT and no glycoproteins, no structural proteins, and these are probably propagated around the genome by the reverse transcriptase that we have. So think about it, our cells are making RT. Any mRNA in the cell can be copied into a DNA, and then it can hop around and integrate in various places. And that's what happens here. These processed pseudogenes are simply DNA copies of mRNAs that we produce, which are copied into DNA by reverse transcriptase, and then they integrate at various parts in our genome. Some of these integrations have pathogenic consequences. There are some human diseases which have been mapped to integrations of some of these retroelements. I'm showing you one of our endogenous retroviruses called HERVK. 8% of our genome is endogenous retroviruses. As I said, all of them are dead, which means they're full of mutations, so they can't make virus. But back in 2007, a group at the Rockefeller took the sequence of one of these endogenous retroviruses called HERVK, and this is the closest to a viable virus that is in our genome. Now that our genome is sequenced, we know the sequence of all these endogenous retroviruses, and they repaired them. They repaired all the mutations, and they got now these particles coming out of cells in culture. So they took the sequence from the human genome, they fixed the mutations in this particular HERVK endogenous retrovirus, and they got virus out, as you can see here. So this is a virus, HERVK, that probably infected our ancestors around 1 million years ago. So here is the speciation of Homo sapiens and its ancestors. So 1 million years would be about right here. So apparently here, HERVK infected us, became endogenized, and eventually mutated so it no longer produced virions. So what they have done is resuscitate this virus. So this is a million-year-old retrovirus back to life, a phoenix, if you will. Who knows what it could do, right? We have one retroviral gene in us, in fact, in all mammals that give rise to live birth, which seems to make a protein, and that is called syncytin. Now, what's shown here is when you get infected with a retrovirus, you can make viruses and transmit them to other people by the conventional routes, but sometimes they get integrated into your germline and then you pass them on to your offspring. That's vertical transmission. So this happened millions of years ago to our ancestors, and most of the genes mutated so viruses weren't produced, but apparently the glycoprotein gene of one of our endogenous retroviruses has been maintained as an open reading frame for all these years. So you don't maintain an open reading frame without mutations unless the protein is doing something. And what we think this is doing is that this syncytin is what it's called now. It used to be called the viral glycoprotein, was captured millions of years ago, and it enabled animals to start producing live offspring because it gave rise to a placenta. So up until that point in evolution, animals couldn't have offspring developing in them because they would be rejected by the immune system. The syncytin allowed fusion to occur in the placenta, in the formation of a placenta, which could separate the mother from fetus. So in every animal you look at, you see evidence for intact syncytin genes, which again are retroviral derived. They're derived from the viral glycoprotein. And just like the viral envelope glycoprotein, when it binds a receptor, can lead to fusion of the virus and the cell, we think that the expression of syncytin, which again used to be an envelope glycoprotein, allows cell-to-cell fusion, and that's how you make a placenta. So again, the idea is, millions of years ago, animals were infected with this retrovirus. They captured the glycoprotein gene, and its expression in the right place allowed the formation of a placenta. So you can imagine that's why it is still expressed today. Now, we can't prove this in humans. I mean, syncytins are expressed in the placenta, but in mice you can knock out the gene encoding syncytin, and those mice do not have placentas. They don't develop properly and they can't have offspring. So good evidence that, in fact, the captured retroviral gene was actually good for us. Now, it turns out that in Australia, the koalas are being endogenized by a retrovirus. So we were endogenized millions of years ago by a retrovirus, by many retroviruses. That's why we have 8% of our genome as retroviruses, but they're not making viruses anymore. Koalas right now are being endogenized. They're being infected, and the virus, it's called the koala retrovirus, is going into their germline. And you may know that, of course, the koala originates in Australia, and the infection has been going from the north to the south of Australia. People are studying this. All the koalas in the north are infected with this retrovirus. There are some in the south that are not yet infected, but it's being spread, and eventually they're all going to have this retrovirus. It does cause disease in koalas. It immunosuppresses them, and it makes them quite sick, so it may end up wiping them out. But in this particular study, they went into a museum where they had collected koala pelts from the 1800s, and they showed by PCR that back then, these animals were already infected with this retrovirus, so it's spread very slowly through Australia. So the reason this is cool is because we can watch endogenization in process. Most of the animals that have endogenous retroviruses, that happened millions of years ago. But now it's happening in contemporary time, and we can watch it spread through a population.